perhaps the most famous pair of identical twins whose story prompted scientists to take a closer look at them. Jim Lewis and Jim Springer were separated at birth, only 4 weeks old, and were raised in different families in Ohio, in homes just 60 kilometers apart. Lewis found out he had a twin brother when he was 5, and Springer was told when he was 8, but they met only once during childhood. Fate brought them together again when they were 39 years old. It turned out that they were remarkably similar. Their wives were both named Linda, and both had divorced, and their second wives were both named Betty. One brother had a son named James Allen, and the other had a son named James Allen. Both worked as deputy sheriffs, drove light blue Chevrolet cars, and enjoyed mechanical work. Both suffered from migraines, smoked Salem cigarettes, drank Miller Lite beer, nervously bit their nails, and vacationed on the same beach in Florida. When Thomas Bouchard, a psychologist at the University of Minnesota, studied their similarities, he declared that their lives were eerily similar. Sometimes their behavior was so identical that even Bouchard was baffled. In addition to taking their fingerprints and conducting various intelligence tests, the subjects behaved the same in almost all other tests, even in terms of having the same health problems. Interesting facts about twins. The frequency of twin births varies from country to country, from 1 to 80 pairs per 140 newborns. There is a higher probability for mothers over the age of 35. In the 6th week of pregnancy, one twin may absorb the other, which means absorbing them. Sometimes, this parasite remains in the body even after the birth of the twin, who is unaware of the presence of this parasite. In ancient times, twins were considered terrifying phenomena. There was suspicion and aversion among gods and heroes when twins were punished. A 15-year study conducted in Germany found that 75.8% of twins are born within 15 minutes of each other, while 24.2% are born with an interval longer than an hour. Case number 2, the Nazi and the Jew. An incredible story of twins raised in completely different cultures. Oscar Stohr and Jack Yuffe were born on Trinidad in January 1933. Their Jewish mother was married to a German doctor, Oscar Stohr. Their father stayed on Trinidad, while their mother took the twins with her to Germany. After a few months, they divorced, and Oscar went to Germany with his mother, while Jack stayed on Trinidad with his father and had no contact. In Germany, Oscar joined the Nazi Party in 1936 and became a loyal member of the Hitler Youth, and later the German Army. In contrast, Jack attended a linguistic high school in Trinidad, studied medicine at Oxford, and later at an Israeli academy. The brothers met again after 40 years in Israel in 1979. During their first meetings, they were shocked to learn they had the same preferences for clothes, even the same gestures and way of walking. They immediately recognized each other and described it as a feeling of unprecedented closeness and belonging. Jack said, "When I first saw him, I knew he was my brother. He wore clothes similar to mine, and when we started talking, we realized we had similar views on many things, even religion, despite being shaped by completely different ideologies." Oscar then stated that the realization of his brother's existence forced him to reconsider his political beliefs and ideological views. Eventually, Oscar became known as a fighter against neo-Nazism, while Jack became a well-known doctor and professor at Tel Aviv University. Genetic heritage has shown that twins, even when raised in completely different conditions, can have incredible similarities. Despite living in 2 different political systems, genetics and heritage played a significant role in their lives. Unexpected results. Since 1979, Bouchard has been dedicated to researching twins separated at birth. He was inspired by the story of the Jim twins mentioned in the text above. Together with a team of scientists, Bouchard managed to find and thoroughly examine about 86 pairs of twins raised in different environments. Of these, 17 pairs were identical twins. Bouchard's team compared these 2 types of twins. "The research was extremely painstaking," said Bouchard. "We found twins in the most diverse places, between city streets. We studied their abilities, personality traits, and social standing. I believe no one has conducted such extensive research before us." It was necessary for the twins to fill out a questionnaire with about 1,000 interesting questions, from their stance on moral values, their fears, aesthetic judgments, to their preferences for certain TV programs and book choices. The answers pointed to different fears they were susceptible to. Every living being exploring biology and sociology questions the concept of similarity, views on life, relationships with parents, and authority. All of these are clear and inherited roots. These results surprised many scientists. Until then, it was unknown how much genetics could influence personal and social attitudes in everyday life. Brent, Johnny, and Mark. In Minnesota, along with Bouchard's team, worked Dr. Nancy Siegel. She, from research titled Virtual Twins, studied children of different ages who, although not twins, lived in the same family. Virtual twins, as explained by Nancy Siegel, already have one child, and they adopt another from an orphanage of the same age. Based on the study, she concluded that these children adopt the same values, behaviors, and habits. Discovering the secrets of evolution. In this zoo, there are elephants, hedgehogs, dolphins, sharks, various fish, snakes, monkeys, and humans, all kinds of animals you can imagine. But this zoo is not ordinary. It is a zoo of genetic data located in Switzerland, in the city of Cava. Each star in this zoo represents a different species with its DNA sequences recorded in their genetic codes. Scientists in laboratories have collected this data to study how species have evolved and changed over time. These studies help biologists identify specific genes responsible for certain traits and functions, allowing them to understand exactly how species have evolved over time. It seems that to explain what is so unique and different about monkeys and what is so similar between mice and humans. Scientists have focused on genes sequences of nucleotides in DNA that contain instructions for proteins that perform specific functions. Researchers have discovered that nearly 99% of all our genes code the same as those in mice. In other words, we humans couldn't function without the genes that make up our brains, hearts, and nervous systems. However, if we have almost the same genes as mice, why do we look and behave so differently? The secret lies in genetic switches, which are like light switches. They turn genes on and off at the right time and in the right place in the body. One of these switches is called an enhancer. Enhancers are DNA segments that help activate genes. They can be very close to or far from the genes they activate. For example, while a gene in mice may be active only in the liver, the same gene in humans can be active in both the liver and the brain. Genetic switches, or so-called enhancers, allow an organism to use the same genes in different ways. For instance, a gene for hair growth in mice might be the same as a gene for nail growth in humans. Differences in gene activation lead to vast differences in physiology, appearance, and behavior among species. Yes, we share genes with fish. We have genes for a swim bladder that helps fish swim. In humans, this gene is inactive. Our genes for facial expressions are very similar to those in cats. Elephants have genes that help them grow large, but mice have similar growth genes that are activated differently. Every gene has at least one enhancer, and some have more. Enhancers help a gene be activated in different parts of the body. For example, genes that code for proteins for hair growth in mice might be the same as genes for nail growth in humans. These differences in gene activation create huge differences in physiology, appearance, and behavior among species. Scientists have discovered that enhancers are particularly interesting because they help understand how genes became active during evolution. Using advanced technologies, they can now precisely map where enhancers are located and how they work in different organisms. One genetic switch consists of enhancers, a short part of non-coding DNA, and so-called transcription factors, specific proteins to which they can bind. The connection with these genes can cover parts of genes that are far from the enhancers or even on another chromosome. The binding or non-binding of a transcription factor to an enhancer determines whether a gene will be turned on or off. When a gene is active, an enzyme is turned on that transcribes RNA copies of the gene. Every gene has at least one enhancer. They are usually hundreds of base pairs long and close to the gene, but they can be thousands of nucleotides away. The most important discovery is that some genes can have more than one enhancer. This is particularly true for genes that code for proteins responsible for the head and body anatomy. The essence is that genes can be activated many times by enhancers in different tissues, increasing the efficiency of individual genes. In theory, enhancers allow specific cells in the body to find their specific genes, as the genes themselves are not equally active in different tissues. In recent years, there have been the first direct proofs that this was a common case in some animals. This phenomenon explains why very different groups of animals can share most of their genes, if not all, which are responsible for building their bodies. Contrary to earlier expectations, it is not about different genetic compositions, but about how and when the same or similar genes are used. Therefore, if we really want to understand why human anatomy differs from, say, that of a monkey, or why an elephant's structure is different from a mouse's, we must accept that most of the data do not lie in our genes and proteins, but in an entirely different realm of the genome that awaits to be explored. If you liked the video, like it and subscribe to the channel to help it grow and spread these smart informations.